Assalamualaikum warahmatullahi wabarakatuh. My name is Ahmad Zarif Razaini from class 4 Ibn Hafiz.
I am Afina Salahabati Syaminti Mu'asafuddin from 4 Ibn Sina.
And one Muhammad Amsyar bin Suhaimi from class 4IN.
Today I will be explaining about cell division and cell cycle. As we all know, cells in our body always grow, divide and die, which means the dead cell must be replaced with new cells. Cells in our body produce new cells through cell division process, which involves two stages, that is karyokinesis and cytokinesis. Karyokinesis involves in the division of nucleus, while cytokinesis involves in the division of cytoplasm. Then the organism body cells are divided into somatic cells and reproductive cells, also known as gametes. Somatic cell is body cells apart from gametes, which is produced through mitosis process. Somatic cells contain a diploid number of chromosomes, which means each cell contains 2 sets of chromosomes and the total of chromosomes is 46 chromosomes. While gamete is a reproductive cell which is produced through the meiosis process. And gamete contain a haploid number of chromosomes, which means each cell contains 1 set of chromosomes and the total of chromosomes is 23 chromosomes. In diploid cells, 1 set of chromosome is from the male parent or paternal chromosome, while the other set is from female parent or maternal chromosome. Both paternal and maternal chromosome have the same structural characteristic. These pair of chromosomes are called homologous chromosomes. Cell cycle is the sequence of events that involve DNA multiplication and cell division to produce 2 daughter cells. Cell cycle has 2 phases, which is interphase and M phase. In interphase, it has 3 phases, which is G1 phase, S phase, and G2 phase, while M phase is made up of mitosis process and cytokinesis. G1 phase is the phase where cells grow. Mitochondrion and endoplasmic reticulum are produced at this stage, and the proteins are also synthesized during this time. The nucleus also looks big, and the chromosome is in the form of chromatin. S phase is where the DNA synthesis occurs. DNA in the nucleus is replicated. Each chromosome will multiply into 2 identical chromosomes known as sister chromatids. Both chromatids contain the same copy of DNA molecules and join at centromeres. G2 phase is where the cells will continue to grow and remain active metabolically. Cells will gather energy and make final arrangements to enter the next stage of cell division. After the interphase stage ends, the cell will enter the M phase. M phase is made up of mitosis and cytokinesis. Mitosis involved in prophase, metaphase, anaphase, and telophase.
Mitosis is defined as the division of the parent cell into 2 nuclei. Each nucleus contains the same number of chromosomes and genetic content of the cell, genetic content with the nucleus of the parent cell. So basically, mitosis is divided into 4 phases. The first phase is prophase. In the nucleus, chromatin starts to shorten and thicken. The chromosome is seen to be made of 2 identical threads named sister chromatids. The sister chromatids are joined at the centromere. Next is metaphase. The centrioles are at the opposite poles of the cell. The spindle fibers maintain the chromosome at the equatorial plane. Metaphase ends when the centromere begins to separate. Right. Next is anaphase. So, what happens in anaphase? The centromere divides into two, and the sister chromatids separate. Then, the spindle fibers shorten and contract, and sister chromatids are attracted to the opposite poles of the cell. Anaphase ends when the chromatid arrives at the poles of the cell. So lastly is telophase. So what happens in telophase is when the chromatids are at the opposite poles, they are now called daughter chromosomes. Each pole contains one set of complete and identical chromosomes. So if you want to remember the steps, if you want to remember the 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 phases, you can remember this step. This step is prophase, metaphase, anaphase, telophase.
To talk about the differences between mitosis and cytokinesis in animal cells and plant cells. Cytokinesis is different between animal cells and plant cells. In animal cells, cytokinesis occurs when the plasma membrane constricts in the middle of the cell between the two nuclei. Microfilaments inside the plasma membrane at the equator pulls the plasma membrane inward. The inward pull on the plasma membrane produces the characteristic cleavage furrow. When the cleavage furrow reaches the center of the cells, it is pinched apart to form two daughter cells. For the plant cells, during telophase, membrane-enclosed vesicles derived from the Golgi apparatus migrate to the center of the cell. Vesicles fuse to form cell plate. The cell plate continues to develop until it connects with the existing cell's plasma membrane. Then cytoplasm divide and two daughter cells form. What is the necessity of mitosis? First, mitosis will produce new cells to replace cells that are dead or damaged when the body is injured. Second, the lizard is able to grow a new tail if the tail breaks through the mitosis process. Lastly, mitosis ensures that rapid cells growth occurs for embryo development. All from us, thank you for watching.
Okay, thank you.
Thank you for watching.